Hi everyone and welcome back to Capsa, your trusted YouTube channel for clear exam focused biology lectures. Today we are going to break down a fundamental but often confusing topic, transport mechanisms across the cell membrane. We will go step by step through the passive, active and bulk transport and even explore real world connections to disease and drug actions. If you truly want to master this topic, whether for exams or deep understanding, be sure to watch this video till the end. You won't want to miss any part. So let's dive right in. Let's begin with the basics of the cell membrane. Think of it as the security gate of a cell, selectively allowing certain substances to pass while blocking the others. This selective permeability is crucial for maintaining internal balance or homeostasis. It helps the cell take in essential nutrients, remove waste products, and respond to external signals, all vital for survival. Now, how do substances actually move across this membrane? The transport mechanisms are broadly divided into two categories. First, passive transport. This process does not need any energy. Molecules move naturally from an area of high concentration to low concentration, just like a ball rolling downhill. Second, active transport. Here, energy is required, usually in the form of ATP, because molecules are moved against their concentration gradient, like pushing that same ball uphill. Now let's explore the passive transport mechanisms. These are the energy-free ways that substances move across the cell membrane. The key principle here is simple. Molecules move from an area of high concentration to low concentration following their natural gradient. No ATP required. You can see in the picture. The first type of passive transport is called simple diffusion. So what exactly is simple diffusion? It's the spontaneous movement of small non-polar molecules directly through the phospholipid bilayer without the help of any transport proteins. Let's break that down. What kind of molecules use simple diffusion? Small non-polar gases like oxygen, carbon dioxide, and nitrogens. Lipid soluble substances like steroid hormones and ethanols also pass through easily. How does it happen? There is no protein assistance needed. These molecules just slide between the lipid molecules of the membrane, simple and direct. Which direction do they move? Always from high to low concentration, or what we call down the concentration gradient. But what controls how fast this happen? Several factors influence the rate of diffusion. First, concentration gradient. The steeper the gradient, the faster the diffusion. Now, temperature. Warmer conditions speed up the molecular movement. Molecular size. Smaller molecules move more quickly. Membrane permeability. A more permeable membrane allows faster movement. What exactly? Classic example: oxygen diffusing into cells to fuel the respiration and carbon dioxide diffusing out as a waste product. No energy, no fuss, just natural molecular motion doing this work. Now that we have covered simple diffusion, let's move to the next form of passive transport, facilitated diffusion. This mechanism is essential for the transporting molecules that cannot pass directly through the lipid bilayer, particularly polar or charged substances like glucose or ions. These molecules need a little help to get across, and that help comes in the form of transporter proteins embedded in the membrane. So, how does it work? Facilitated diffusion still moves substances from high concentration to low concentration, and importantly, it still does not require any energy. But instead of slipping through the membrane directly, these molecules rely on two types of transport proteins: channel proteins and carrier proteins. Channel proteins create water-filled pathways or hydrophilic pores that allow certain ions or water molecules to pass rapidly. For example, ion channels allow sodium, potassium, or calcium ions to move in and out of the cell. Many of these channels are gated, meaning they open or close in response to electrical or chemical signals. Another example is aquaporins, specialized channels that dramatically increases water movement, especially in cells like those in the kidney or red blood cells. Now, the carrier proteins, on the other hand, operate a bit differently. They bind to the target molecule, such as glucose, and then undergo a shape change that carries the molecule across the membrane. A well known example is GLUT transporter, particularly GLUT1, which helps glucose enter the red blood cells. So, what makes facilitated diffusion unique? Its specificity. Each transport protein is selective for a particular molecule or group of molecules. It also shows saturation behavior. When all transport proteins are busy, the rate of diffusion reaches a maximum, no matter how much solute is outside the cell. And lastly, many of these channels are regulated, especially the gated ones, allowing the cell to control when and how substances move. A particular example of this in action is when glucose enters a red blood cell through GLUT1. Glucose, being a polar molecule, cannot pass through the hydrophobic core of the membrane on its own, but thanks to carrier proteins. It transported efficiently, again, all without using any energy. So, facilitated diffusion is an elegant example of how cells combine selectively, efficiently, and control all while the conserving energy. Now let's turn our attention to a very special type of passive transport, osmosis. So osmosis is all about movement of water, especially it's the diffusion of water across a selectively permeable membrane, moving from an area where solute concentration is low to where it's high. In simpler terms, water moves to dilute the more concentration side. So it's the cell's way of balancing the solute levels on both sides of the membrane. Here is the key idea. While solutes like salts or sugars often cannot cross the membrane freely, water can. So instead of solute moving to balance things out, water moves instead, flowing towards the side with more solutes in an effort to dilute it and restore the balance across the membrane. The result? A pressure buildup, and that's known as osmotic pressure, the force required to stop water from moving across the membrane. Now, how does this affect the cells? That brings us to an important concept, tonicity, which describes how a surrounding solutions affects the cell volume. Let's break it down into three types of solutions. So first is in an isotonic solution, the solute concentration is equal inside and outside of the cell. There is no net water movement. So the cell stays the same. For animal cells, this is ideal condition. They remain stable and functional. In plant cells, though isotonic conditions make the cell a bit soft and flaccid because there is no extra water pushing against the cell wall. In hypotonic solution, the solute concentration is lower outside of the cell. Water rushes in. For animal cells, this can be dangerous. The cell may swell and even burst, a process called lysis. But for plant cells, this is ideal. Water fills the central vacuole, pushes the cell membrane tightly against the wall, making the cell turgid and structurally strong. In contrast, a hypertonic solution has a higher solute concentration outside the cell. Water moves out, causing the animal cell to shrink or crenate. In plant cells, the membrane pulls away from the cell wall, a process called plasmolysis, which leads to wilting. So, is there any real life example or real world example? Yes, IV fluids given in hospitals are carefully prepared to be isotonic because if they were too diluted or too concentrated, red blood cells could either burst or shrink, leading to serious complications. In plants, osmosis is what maintains the target pressure, helping the plant stand upright and stay healthy. So, osmosis may seem simple, but it plays a vital role in hydration, cell shape, and survival of both animal and plant cells. Now that we have covered how substances move passively, let's shift gears and look at active transport mechanisms, processes that require energy to move substances against their concentration gradient, that is, from low to high concentration. This is like pushing a ball uphill. To what they need, cells actually need energy, usually in the form of ATP, to make this happen. Let's begin with the first type, primary active transport. In this mechanism, just the cell directly uses ATP to fuel the movement of molecules across the membrane. The most classic and essential example of this is the sodium potassium pump, also known as sodium potassium ATPase. Here is how it works. Inside the cell, the pump first binds to three sodium ions. Then, using energy from ATP, the pump gets phosphorylated, meaning a phosphate group is added to it. This causes the protein to change the shape and release the sodium ions outside the cell. Next, it binds to potassium ions from outside of the cell, and after releasing the phosphate group, a process called dephosphorylation, the pump returns to its original shape and releases the potassium inside the cell. So, in one full cycle, three sodium ions are pumped out and two potassium ions are brought in using one ATP molecule. So, why is this important? This pump plays a critical role in maintaining the resting membrane potential, especially in nerve and muscle cells, allowing them to generate and transmit electrical signals. It also creates the ion gradients that are necessary for many other transport processes, including secondary active transport, which we will look at the next. And this is not the only example. Other primary active transporters include calcium pumps, which move calcium ions out of the muscle cells to help them relax after contraction. Proton pumps found in the stomach lining, which actively secretes hydrogen ions to produce the gastric acid. Let's now explore the second type of active transport, secondary active transport, also known as coupled transport. Unlike primary active transport, this mechanism does not directly use ATP. Instead, it harnesses the energy stored in ion gradients, usually sodium, that were created by primary active transport system like sodium potassium pump. So think of it this way. The sodium potassium pump spends ATP to build up a high concentration of sodium ions outside the cell. Okay? That sodium wants to rush back in, down its concentration gradient, and secondary active transport uses that flow like a water wheel, capturing the energy of sodium movement to carry other molecules along. Now, depending on how the transported substances move in relation to each other, there are two main types of secondary transport, symport and antiport. In symport, also called as co-transport, both molecules move in the same direction, one down its gradient and the other against its own. A great example is the sodium glucose symporter in intestinal cells. Here, sodium flows into the cell naturally, and glucose hitches a ride with it, entering the cell even though its concentration is already higher inside. This is how our intestine efficiently absorbs glucose from digested food. In contrast, antiport or exchange transport involves two molecules moving in opposite direction. A classic case is the sodium hydrogen exchanger found in the kidney cells. Sodium moves into the cell down its gradient while hydrogen ions are pushed out, helping to regulate the blood pH by removing excess acidity. In both the cases, the key energy source is not ATP directly, but rather the sodium ion gradient that was set up by the ATP powered sodium potassium pump. So, even though no ATP is used during the coupled transport itself, it still relies heavily on the energy invested made by primary active transport. So, secondary active transport is a clever strategy cells use to maximize efficiency, allowing the movement of essential nutrients and ions by simply riding the wave of existing gradients. Now let's look at the fascinating category of transport mechanism, bulk transport. Unlike passive or active transport, which move individual molecules or ions, bulk transport handles large substances like proteins, polysaccharides, or even entire cells by using membrane bound vesicles. This is how the cells import or export large quantities of material in one go. We will divide this into two main processes, endocytosis, exocytosis. Endocytosis, which is called as import into the cell. So endocytosis is the process by which the cell membrane folds inward, wrapping around substances in the external environment and enclosing them within a vesicle. This vesicle then bursts up into the cell, bringing the material inside. There are three main types of endocytosis, each with a unique role. So, phagocytosis or cell eating is used by the cell to engulf large particles such as bacteria or cell debris or cellular debris. The material is surrounded by a membrane enclosed in a vessel called phagosome and then usually fused with the lysosome for digestion. This process is especially important in our immune system. For example, macrophages use phagocytosis to devour the pathogens and keep us healthy. Next is the pinocytosis or cell drinking. In this case, the cell takes in extracellular fluid along with any dissolved solids it contains. Unlike phagocytosis, pinocytosis is non-selective. The cell samples whatever is around it. This allows the cells to absorb nutrients and maintain the fluid balance. Then we have receptor mediated endocytosis, a much more specific and efficient method. In this process, the cell uses receptors on its surface to bind to specific molecules or ligands. Once each ligands are bound, the membrane folds in, forming a vesicle loaded with the targeted substances. A key example is the uptake of LDL cholesterol. If this pathway is defective, as in familial hypercholesterolemia, cholesterol levels can rise dangerously in the blood. Exocytosis. Export out of the cell. The opposite of the endocytosis is exocytosis. Here the cell packages materials into vesicles inside the cytoplasm, which then fuse with the plasma membrane, releasing their contents to outside the environment. This is how cells communicate and secrete important substances. For example, neurons releases neurotransmitters like acetylcholine at synapses via the exocytosis process. Pancreatic beta cells release insulin into the bloodstream to help regulate the blood sugar. Now, truly appreciate the importance of membrane transport, we have to look beyond the cell and into the clinic. Because when these transport mechanisms malfunction, the consequences are far reaching. One of the clearest example is cystic fibrosis, a genetic disorder that arises from a defect in the CFTR chloride channel. Normally, this channel helps maintain the proper balance of salt and water across the epithelial surface, but when it's faulty, chloride ions cannot exit the cell efficiently and water does not follow. The result is thick, dehydrated mucus that clogs the airways, impairs the lung function, and disrupts enzymes flow in the pancreas. So, this is not just a textbook defect, it leads to renal breathing difficulties, chronic infections, and digestive problems. If you want more about it, I have a video. You can see the playlist Disease, Disorder, and Syndrome. Another example comes from a condition that affects millions globally. Diabetes mellitus. In healthy individuals, insulin stimulates the movement of glucose into cells by activating specialized glucose transporters like GLUT4. But in diabetes, particularly type 2, this insulin mediated transport is impaired. Either the body does not produce enough insulin or the cells become resistant to it. As a result, glucose remains trapped in the bloodstream, leading to persistent hyperglycemia and a wide range of complications involving the eyes, kidneys, nerves, and heart. We also see the impact of transport dysfunction in the familiar hypercholesterolemia, a genetic disorder where the LDL receptor, which normally helps clear bad cholesterol from the blood, is defective or absent. This leads to abnormally high cholesterol level and a greatly increased risk of early onset atherosclerosis and cardiovascular disease, sometimes even in young adults. But the relevance of transport mechanism does not end with the disease. They also serve as critical targets for some of the most widely used medications. For instance, proton pump inhibitors like omeprazole are prescribed to treat the acid reflux or gastric ulcers. These drugs work by blocking the hydrogen potassium ATPase pumps in the stomach lining, reducing the amount of acid secreted into the gastric lumen. Similarly, calcium channel blockers like verapamil helps control the high blood pressure by limiting the influx of calcium into the heart and smooth muscles, reducing contraction strength and relaxing the blood vessels. In the nervous system, these transport systems plays an even more fundamental role. The sodium potassium pump is absolutely essential for maintaining the electrical gradient across the neuronal membrane, a requirement for generating action potential. Without this constant energy driving pumping of sodium out and potassium in, neurons would lose their excitability. And when it comes to communication between neurons, exocytosis takes center stage. Neurotransmitters like acetylcholine are packed into vesicles, and when a signal arrives at the nerve terminals, these vesicles fuse with the membrane and release their contents into the synapse, allowing the message to jump from one neuron to the next. In short, the transport mechanisms are far more than microscopic shuttles. For ions and molecules, they are fundamental drivers for health, physiology, and medicine. From genetic disease to drug development, from nerve signaling to hormone release, they underscore the complexity and the coordination required to keep our bodies in balance. So, understanding how substances move across the cell membrane is not just about passing an exam. It's about understanding how life works at its most fundamental level. These mechanisms power everything from breathing and thinking to fighting infections and responding to medication. That is the beauty of biology. The tiniest system have the biggest impact. If you found this lecture helpful, don't forget to subscribe to this free educational platform. It costs you nothing, but it helps us keep creating quality content just for you. And if you have friends or classmates who could benefit from clear, exam focused explanations like this, share this video with them. Learning is better together. Thanks for watching. Keep exploring, keep learning, and I will see you in the next lecture.